Well, welcome back, everybody. This is module 3.4 on the CompuCell workshop. It's going to be an introduction to network modeling inside of CC3D. I remind you that the lectures are being live streamed and recorded, so please be aware of that. Network models are a very large topic, and in fact, Herbert Sauro 2 weeks ago spent an entire week teaching the subject. I know some people in the class have taken that course. Others have maybe learned this material in other ways. I'm going to talk very briefly about a few issues that you might want to be aware of when you're doing network models. And then we're going to focus on how to build network models inside of CompuCell. And so I'm going to cover this in a bit of a hurry. And there are going to be a lot of issues that are of interest and importance that we're not going to be able to go into in detail now. I'm also going to say in advance that the particular simulation that we're going to use in this module has some concepts and materials in it that we're not going to cover right now and that Josh will cover later in the day. So I'd ask you to focus on the network part of the simulation and not worry right at the moment too much about how the back-end magic of the simulation works. So we're talking, this course was always called multi-scale modeling, and we've seen slides like this before. The body has many, many levels of organization from individual molecular reactions, for example, in metabolism, through the macromolecular machinery and transport that goes on inside of a cell, through the organization of cells into tissues, tissues into organs, macroscopic transport, for example, in the blood and the lymph, or in air, in the lungs, and then large transport by blood, for example, between organ systems. And so we have always many, many scales to think about. Typically in CompuCell, we're working at the level from subcells to cells to tissues. We're neglecting the complexity of macromolecular machinery and a lot of the structure within cells. Although yesterday we saw a simple model where the cell was broken up into 3 compartments, a nuclear compartment, a lamellipodium compartment, and a cytoplasmic compartment. And one can go a lot further in that kind of development if one wants to. I'd like to say again that this is going to be a very quick overview of some very important concepts. I strongly recommend the manuals that Herbert has written for tellurium antimony. The tellurium Read the Docs has a great tutorial with online executable code for doing simple network simulations. I've mentioned before Herbert Sauro's textbook, Systems Biology and Introduction to Pathway Modeling. That's a very elementary book, but covers step-by-step in real detail how to start building these kinds of models. And we have in our Google Drive the tellurium antimony cheat sheet that gives you the basics. And again, I encourage people to print that out and have it on your desk to remind yourself of the basics. Although I have to say the antimony language in some ways is so simple. The reason we use it is because of that simplicity. It really makes it easy to build this kind of model. Knowing what model to build is not necessarily so easy, but implementing it is. Farouche asked, could you please send the link to the live stream? Okay, let me do that. Does somebody else have it? Okay. There's the live stream link. Okay. And if you want to learn more about the topics that we're only going to be touching on today, there are many, many textbooks and places that you can go. These topics would include things like dynamical systems, the theory of stability of networks and fixed points of networks, verification theory, There are books on how to calculate networks and infer networks from experimental data. And there are wonderful books on mathematical biology of all kinds. And again, I could make some suggestions offline if Herbert's textbook isn't the one you want. Herbert's book has some very good references in it as well. We're just going to be talking about some very simple ideas today, and I'm going to skate over a lot of the complexity because the goal here is to get you writing these models. If you've done this kind of modeling before, I hope you'll find it relatively easy to do it. If you haven't done it before, I hope the examples are simple enough that we can work through them together. So when we think about biological networks, there are four main kinds of biological networks that we're going to deal with. There are chemical networks and metabolic networks where we are creating and destroying molecular species. And the classic example of those would be metabolism in the mitochondria. Those tend to operate on very fast timescales. There's signaling networks that transfer information within and between cells, things that would involve molecular receptors on the cell surface, G proteins, and so on, kinases. Those tend to operate on a slightly slower timescales than the metabolic networks. They're regulatory networks that turn gene expression on and off or do post-transcriptional modification. Those operate typically on slower timescales still, timescales of 15 to 20 minutes. So molecular metabolic networks, we're typically talking about tenths of a second or less. Signaling networks, we're talking about seconds to minutes. Regulatory networks typically operate on a timescale of 15 minutes and above. And then there is a whole separate question of building models of macroscopic transport, which are used in things like drug discovery. And those are physiologically based pharmacokinetic models. And those typically have a mixture of timescales. Blood transport is very fast, but things like elimination of chemical species are quite slow. And they have their own world. The mathematics is the same, but they have their own world about how to do parameter fitting and so on. But all of these models, all of these networks can in fact be implemented computationally in the same way and mathematically in the same way. These networks tend to operate together. Excuse me if I'm drinking about a little hoarse this morning. And so we want to ask a lot of questions about how networks operate. We can ask the question, does the network that's active inside of a cell change? When a gene is turned on or off, you have molecular species that can interact that weren't there before. When you have a cell that has a particular set of networks active, the state of the network could change. You could have more of a phosphorylated version of a protein or less. And of course, the components of the cell themselves could change because you could build additional mitochondria. You could change the structure of the cell. And typically, we have to think about all of these networks together. However, very often in the simulations that we're doing, we're going to assume, just the way when you drive a car, as long as the car is working properly, you don't worry too much about how the engine works. For many of the things that we're going to do, as long as the cell is alive and doing whatever it does normally, we're not going to worry so much about metabolism. If we're interested in the production of toxins by as a metabolic byproduct, then we'll model that. But the basic metabolism of the cell is something we typically have hidden under the hood, so to speak. So if we talk about chemical reactions, it's interesting. I'm sorry, PowerPoint has hidden a slide for hidden a figure for me. I apologize. We are talking about transformations, chemical transformations. If we think about a chemical reaction formula, here 2-ADP go to ATP plus AMP. That's a reversible chemical reaction, or some kind of system of chemical equations. 2 molecules of A go into B. B can make 3 of C. C and A can combine to make D, and these are reversible, what we'll see is that we're used to writing chemical reactions in the form of arrow diagrams. These chemical reaction formula are in the form of a substrate on the left and a product on the right. We get rid of 2 ADP and we get 1 ATP and 1 AMP. On the other hand, we could run this backwards and take an ATP and an AMP and turn it into 2 ADP. We're not talking about here about the energy. So then when we think about a network more generally, we have to think about the nodes. Those are the values that sit, A, the amount or concentration of chemical A, the amount or concentration of chemical B, the amount or concentration of chemical C, and arrows. And in this case, these arrows represent chemical transformations. We destroy A and create B. We destroy B and we create C. We combine A and C, which destroys them, and create D. Now, of course, chemical reaction conserves mass, but the key thing to think about in a diagram of this kind is that we have A goes to B, and it has a rate, and these are called rate laws, and the rate tells you how fast A is used up and B is created. Classically, in simple chemical reactions, the simplest form of chemical reaction rate law that we have is called the law of mass action, which says that the rate at which we take A and B and produce C is a constant times the amount of A times the amount of B. That is, it's bilinear in A and B. At least for the case of not having a lot of A and B, that makes sense. If we don't have any A, we can't produce C. If we don't have any B, we can't produce C. And if we think mathematically in terms of a Taylor expansion, the lowest order term is going to look like that. Now, when we have very high concentrations of A and B, typically we'll get some kind of saturation, but at least at low concentrations, mass action works pretty well. And there are whole textbooks which assume everything in the world is mass action or a simple generalization of that. It makes the mathematics a little bit easier. But one thing to remember is that this one arrow diagram, A plus B goes to C at a rate VF of AB, actually generates 3 differential equations. One for the amount of the concentration of A, one for the concentration of B, and one for the concentration of C. And if you're used to writing ODEs for rate laws, you have to remember that when you start writing rate laws in terms of arrows, the number of equations that you write is slightly different and their form is slightly different. And we're going to be writing things in terms of these arrow diagrams in antimony. The natural language of antimony is arrow diagrams. Now, in reality, life is more complicated. Metabolic networks are usually segregated in space. And within the cell, there are all sorts of complicated structures like the endoplasmic reticulum and the Golgi apparatus, which subdivide the cell into compartments. We're typically going to assume that, as most network models do, that the cell behaves like a stirred reactor, and we're going to neglect that spatial compartmentalization unless we're going to model something very specific. And so we're not going to think about spatial structure to begin with. Although in the example we showed yesterday, we could perfectly well have different chemical reactions happening in the nuclear compartment, the cytoplasmic compartment, and the lamellipodium compartment. The next kind of network that we have to deal with are signaling networks. And a classic example of this would be Notch. Notch is a transmembrane receptor. When it binds on another cell to delta, it's cleaved, and the intracellular component of Notch then goes into the cytoplasm of the cell and sends a signal that, among other things, can control cell proliferation. Another example of this would be the classic phosphorylation kinase cascade of MAP kinase, We start out with map kinase, 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 which phosphorylates map kinase, kinase, which phosphorylates map kinase, which phosphorylates a bunch of other stuff and does things. And so that kind of kinase ladder is something that we'll model a lot. Signaling networks are things that we're going to want to be able to model. And one of the things that we have to think about in a signaling network diagram is that the signaling networks don't have the same transformational structure. The arrows mean something different. And I'm going to talk a little bit about arrows just to help people out, because there was a request for that from somebody before we started the course. The kinds of questions you might want to ask about a protein signaling network would be something like, if I increase the amount of vascular endothelial growth factor, VEGF-A, in the environment of an endothelial cell, vascular endothelial cell, does the cell proliferate or does it die? Depending on whether it's a physiologically meaningful concentration, you could get both responses. What happens when the number of receptors on the cell membrane changes? How does that affect the cell's migration? If I mutate a component of this network, what happens? And if I mutate that component, does that lead to a change in the cell behavior that leads to a disease state? That's very important if I'm thinking about cancer. A note about networks, protein signaling networks. Typically, you'll see these lollipop diagrams, lollipop arrows for activation, these T-shaped arrows for inhibition. We'll use the pointed end arrow for mass transformation. And links come here. Typically, we're talking both about chemical transformation. When a molecule is phosphorylated, that's a chemical transformation. But the kinase that's doing it may or may not be molecularly, well, if it's a kinase, it actually is reacting because it's transferring the phosphate. But in many cases, we'll have a situation where we'll see a diagram like the one on the top right, where we have S1 is transformed to S2. S1 could be the non-phosphorylated form. S2 could be the phosphorylated form of the molecule. And A here is promoting that phosphorylation. In this case, it's a regulatory arrow. A is not being used up in this reaction. Similarly, we could have R, which is inhibiting this transformation. And regulatory links don't use up typically the components. And we have to think about how to represent that. And what we have to think about then is if we have S1 being turned into S2, and that's promoted by A, what that means is that the rate of production of S2 increases as the concentration of A increases. Sometimes this diagram, A promotes the transformation of S1 to S2, is written with a shorthand that just says A promotes S2. You'll see that a lot in biology textbooks. When we see that, we have to interpret it in the following way. We don't know what was being used up to produce S2. And so we have to think about this diagram, A promotes S2, as nothing, some unknown thing, produces S2 at a rate regulated by A. As a chemical reaction, that means nothing goes to S2 at a rate that depends on A. Similarly, if R inhibits S1 going to S2, that's sometimes written with shorthand, R inhibits S2, that's interpreted as a chemical reaction, nothing goes to S2 at a rate regulated by R. And as a chemical reaction diagram, that is nothing goes to S2 at a rate V1, which is a function of R. And so we'll see a lot of what are called dangling arrows, where we either have nothing going to a chemical species or a chemical species going to nothing. It's not very different when we talk about gene regulation, which describes how genes are turned on and off. There are these standards for how to diagram gene regulatory networks. They make things look very simple. In fact, they're not as simple as these diagrams suggest. Gene regulatory networks typically control long-term changes of cell behavior. especially things like cell differentiation, although they're not the only thing that happens in cell differentiation. And so we could ask the question about, if we change the amount of a particular protein, how does that change the amount of messenger RNA being produced? And then downstream, how does that change the amount of the protein coded by that messenger RNA? What happens when we knock out a gene or knock in a gene or make a particular mutation to a gene? Does a network have a stable state which we could describe as a cell type? Does a network allow for differentiation? And of course, very often we're interested in whether drugs change the gene expression in these kinds of networks. Sometimes we're also interested in toxicity. A lot of toxicity studies look at the effect of particular molecular species on gene regulatory. The typical diagram for a gene regulatory network looks like this. Here we have a molecule I, it's an inducer. It acts on gene P. Gene P produces messenger RNA, and the messenger RNA is then translated to protein. Notice that I is not used up in this. The gene is not used up in this. The messenger RNA is not used up in this. And the protein is not a product directly of the messenger RNA. It's produced by the ribosome. And there are a bunch of steps involved. The inducer has to bind to the gene. The gene has to be transcribed to messenger RNA. Well, transcribed to RNA. The RNA has to be edited to messenger RNA. The messenger RNA has to be transported to the ribosome. The ribosome has to translate the messenger RNA to the raw protein. The protein has to fold, and then there's post-translational modification. So there are a whole series of steps that are hidden in a diagram of this kind. Very often that mRNA is hidden as well, and we might even see just I promotes P without going through those steps. If we want to think about translating that, we'll see in a book, P1 promotes P2. P1 is some kind of transcription factor. P2 is a protein. What's really happening is P1 binds to a regulatory site on the gene. The gene is transcribed. This would be is transcribed and then translated to produce P2. As a chemical reaction, the things that are used up are not modeled. So we have nothing goes to P2 at a rate that depends on the amount of P1. Very often we want to think about the fact that the protein will decay. And so we'll also imagine usually a first order decay kinetics where the protein turns into nothing again. So this simple diagram, P1 promotes P2, actually turns into nothing, turns into P2, and P2 goes back to nothing. And what's important are how these rates depend on the amount of P1 and the amount of P2. If we wanted to include the messenger RNA step, because that has a significant delay, then we would have the promoter binds to the transcription factor promoter binds to the activation site. We have transcription to messenger RNA. The messenger RNA itself can decay. The messenger RNA can be translated to P2, and P2 can decay. So this diagram, P1 promotes P2, actually then translates into 4 reactions with 4 rate laws. And so that's something that we always have to remember when we're going from the biological description to the mathematics. Typically, when we are going to interpret P1 promotes P2, we don't know the details of how P1 turns P2 on. Sometimes we'll use Boolean models of that. In continuum ODE models, typically we will assume a Hill function, a sigmoidal rate of production of P2 as a function of the amount of P1, with a basal rate of production, B1. And the steepness of this curve is controlled by this exponent n, and the half maximum concentration for P1, at which we produce at half our maximal rate, is defined by a Hill constant K1. We'll use typically the balanced form where we exponentiate K1 with the same exponent as P1. The steepness of this curve is determined by the cooperativity of the reaction. That could typically be 4 or maybe 8 for steep reactions, more Boolean networks. One way, if you want to build these in a consistent way, is to always rescale reactions between 0 and 1. In that case, K1s are always set to a half. And you can have increasing functions for activation. If P1 inhibits P2, the nice thing about Hill functions is that all you do is you change the numerator from being P1 to K1, and now you get a decreasing sigmoid with the same half maximal value, and the functional form looks the same. And so if I were going to turn P1 into P2, then if P1 inhibits P2, you will get an ODE that looks like the rate of production of P2 is some basal rate of production with a decreasing Hill function, and then P2 decays. Gene regulatory networks come in motifs. There are a lot of classical motifs, Herbert's textbook, and there are many textbooks that talk about some of these. The simplest ones are gene activation or repression of a single gene, but you can have 2 activators or inhibitors that act together. You have to then think about how they combine. You can have a gene cascade where transcription factor 1 turns on gene 1, which produces another transcription factor which turns off gene 2. You can have autoregulation where a gene produces a transcription factor that turns itself off. The effect of those transcription factors can be modulated by other molecules, and they're very, very often regulated by phosphorylation. Typically, because of the time it takes to do transcription and translation, that's typically 20 to 40 minutes for a typical protein. These networks operate on a fairly slow time scale. If we now jump to the blood transport in the whole body or lymph transport in the whole body, these are now physiologically based pharmacokinetic models. These are also network models. In this case, we're modeling flow. Typically, what we'll do to begin with is draw compartments, and these would represent main organ systems: the brain, the kidney, the heart, muscle, fat, other tissue. Here's the liver broken out, the gut, the lung, and the blood as these 2 compartments, the venous blood and the arterial blood as separate compartments. In this case, the basic model structure is a transport equation that says how much material the blood is carrying at any given time between the heart and the venous blood, or between the venous blood and the lung, and so on. And we might also want to know how molecules are being absorbed, eliminated, or absorbed by these various components. And these typically would be asking questions like, if I take a pill at a time X, how long does the pill take to get into my bloodstream? How long does it take to be eliminated from my bloodstream? What fraction of the molecule in the pill goes to particular organs and particular targets? Numerically, these are fairly simple models. We have these fluxes Q that describe the transport between the various components. In this case, there's an explicit picture where the kidneys are excreting the molecular species into the urine and eliminating it. Here, the gut lumen, your intestinal contents, are being absorbed by the gut. The liver is breaking down the molecular species, putting them out in the bile for excretion in the feces. This kind of structure is also a network. Molecularly, these are quite simple. You have typically what are called Fichian laws, where you have transport, forward and backward transport, that's proportional to the concentrations. And in the case of these kinds of models, you typically actually convert concentrations to amounts. You have to know how much the volume of the blood is, the volume of your various tissue and organs are to do that conversion. But again, you can write everything in terms of an arrow diagram. Blood, molecular species in blood is being transformed into a molecular species in body. Molecular species in body is being transformed to molecular species in blood. So all of these kinds of arrow networks can be written in terms of arrow diagrams. Of course, in reality, all of these things work together. The cells wouldn't be alive if they weren't doing metabolism. They're continually receiving signals and sending signals to the outside world and signaling within the cell. Their gene activity is continually changing. And of course, to supply the metabolism of the cell and to get rid of the cell's waste products, we need transport through these PBPK networks. And so that's lightning fly-through of what would probably take a week or two to go through in a regular course. But I'm assuming that people have had some familiarity with this kind of work. On the other hand, you may, if you're coming from a biological background, not have thought so much about how to translate those arrows, which are the classic language biologists use to represent these models, how to translate those arrows into mathematics and then into code. So I did want to show you a couple of rate equations so that you have an idea of what they look like. Okay, so now let me see if this will work for me. I'm going to now split screen. Can people see both screens okay? No.
No.
Okay, let me reshare. Thank you. How about now? Okay, let me do a quick poll. I meant to do this poll earlier, but I guess it's a little bit late, which was to ask people how much network modeling experience they had. I meant to do that at the very beginning of the class. While we're doing the poll, please download the example from the 3.4 folder. If anybody is having any trouble finding that example, please reach out to Giuliano or Jim Sluka and get some help, because if you can't do it, it's going to be boring for the next half hour. And then if you're using NanoHub, please upload it to NanoHub. I will walk you through that, but it would be helpful. Okay, so if I look at this, the responses to my questionnaire, a few people haven't answered yet. It looks like about half of the people have done some kind of network modeling, and about half of the people haven't. And so my guess is that if I do a poll on the module, the people who said that they'd done network modeling before will say I went too slowly, and the people who haven't done it will say I went too fast. For both sides, I hope for the people who have done it before, there was some material in there that was presented differently. And for the people who haven't done it before, I ask you to hang in there. As I said earlier in this class, think if something's worth hearing once, it's worth hearing twice. And so you'll hear these things again. And so nothing else, I hope you got pulled away from the presentation. A few key concepts of different kinds of networks and the idea of a rate equation and the idea of using chemical transformation diagrams to represent different kinds of biological network. Okay. Oops. Okay. I don't know why Zoom decided to change my screen there. Okay, so now what we're going to do is I'm going to do a very, very simple introduction to writing these kinds of models inside of CompuCell. And then in the afternoon or later today, Josh will work in much more detail about how to do this. We will use a specification called Antimony to define these models. And we will then convert those models into something that's executable. In this lecture, we are not going to worry about how that loading is done and how that conversion is done. That will be left to the afternoon. Again, on tellurium and antimony, please go to the tellurium, read the docs, and please do print out those handouts, those cheat sheets that we sent you earlier. Okay, so the first thing that I need people to do if you're using NanoHub, if you're not using NanoHub, you don't have to do this. It's easy. Once you've downloaded the file, you're done. But if you are using NanoHub, please go to the demo folder, download that demo, and then use the Jupyter package or the Tellurium package, whichever one you like, to do the upload. So launch Jupyter Notebooks, and then once you've launched Jupyter Notebooks, hit upload. Once you've hit the upload, find the zipped file that is the demonstration. It's called Tellurium. And then it will ask you to do confirm the upload. And then once you have done the upload, please switch inside of NanoHub to CompuCell 3D v4 main tool. If you're using this on your desktop, simply open Intuit the file tellurium.zip. And I'm going to be checking because it's critical before I go on that everybody have that. Okay, I'll wait just another minute or two for people to catch up. If you're having problems, please reach out in the chat. The demo is called tellurium.zip. It is in the folder module 3.4 exercises. Is anybody else having trouble finding it? And yes, you have to upload it exactly the way we've done it before. Looks like 26 people have got it. Let me do it again. And just make sure that everybody's okay on this. Can I ask the people who are needing more time to please reach out to one of the instructors for help? Please join a breakout session and get some help. If you're running on desktop, all you have to do is download that file and open it in Twitter, so you can skip the next couple of steps. Somebody asked, where is this file tellurium.cc3d? Please reshare. Giuliano, could you please reshare the link to that file? Tarim, thank you very much. Somebody asked for help. Polly, thank you very much for posting the links. Appreciate that. Could somebody join a breakout room? Jim, could you possibly join a breakout room? Is it Igniola needs some help? Would you be willing to take them to a breakout room? You should have access.
Sure. What was the name again?
I will.
Okay, I see it. I see it. Okay.
Does anybody else need help? I don't want to leave people behind, but I also we've got a lot to cover. And so I want to make sure that, but if you can't get this to run, it's going to be boring. Okay, looks like I can move on. Okay, so now I want you to go to CompuCell, switch back to CompuCell. So please switch back to CompuCell. NanoHub seems to have clobbered my CompuCell. So let me go back to CompuCell here. Okay, is anybody having trouble? I seem to have a little bit of an issue with the screen. Does anybody having trouble launching CompuCell? So come back to CompuCell. Inside of CompuCell, select Open Project. Here I'm in Twitted. So actually, if you're in, you can do it from either Twitted or CompuCell, but launch Twitted is probably easier. And you should go to your directory, and there will be a folder called tellurium.zip. When you open that, it will ask you to save it, to unzip it. It may give you a warning that it doesn't have a unique name. Once you've unzipped it, you have to go back to the open and select open again. I've already unzipped it, so if I unzip it again, it'll throw an error. So I'm not going to do the unzip myself. But again, here you'll get this window. Select tellurium.zip, hit open. Which I'm not going to do because I've already unzipped it. If it gives you an error, throws a warning, it should still unzip it, go back, select the unzipped module, which is now called Tellurium. And it may be hiding one level down, and select CC3D, tellurium.cc3D, and you should be able to open it. So I'll wait until everybody has that running. If you're doing this on your desktop, open tellurium.zip in Twitted, unzip it, and launch it. So let me make sure everybody has it launched. Okay, everybody's got it. Okay, good. I know it's not the most convenient thing in the world, but people will get used to it. It's not so bad once you've done it once or twice. Okay, so everybody says they've got it. Then once you have it, click on, actually I have multiple copies of it, don't I? The one that I have here is this one called Simple Infection Model. Okay. So you can, I'm sorry, let me find the one. Sorry, I shouldn't have had multiple ones open. This is the one, no, which is the one. I have to find the one Josh wrote. Oh, here it is, the bottom. Here it is. This is what you should have. Let me do a little screen rearranging here so we can see better what we've got. Okay. and double click on Tellurium CC3D, select Tellurium Steppables. Okay, make sure everybody's got that. And go to the top of Tellurium Steppables and you should see this. I'm going to maybe change the size of my window a little bit here so I can see better. So I want one more line. There we go. So I hope everybody has this on their screen. And as soon as everybody has this on their screen, we can now talk about how to write a network model. And again, there's some magic under the hood, not very complicated, but I don't want to talk about that now. I would ask you to save your questions about how this little simulation works until Josh's explanation this afternoon. What we have here in this code is a place where we can type the model that we're going to run. And then in line 18, something called variables that says which variables from the model we're going to plot and what color we want them to be. And so this is a very simple code in CompuCell that allows us to take a computer simulation written in Antimony, run it and plot it. And we're going to focus today on the Antimony, the language that we use. And we're going to notice if I look at this, that it's written in a string. Here, model equals triple quotes. That's Python's way of saying that the string has carriage returns in it. And a list of the variables. So here we have variable X. Here we're going to say we're going to plot X in red. We're going to plot Y in blue. And we're going to see in line 6 and 7, something that looks like a chemical reaction. J1, the name of the reaction, colon, X transforms into Y, semicolon, rate B times X. And that's all you need to know about Antimony. This is how you write. You can write ordinary differential equations. You can write chemical reactions and so on. So why don't we actually just run this simulation? So click on Tellurium 3D, and now you can either use the project buttons to run it in Player, or here I will run it in Player myself. And I have to switch to the Player. And I may have to re-lay out my screen a little bit. And so here it's misdrawn, so I have to do my usual joggle of the corner to get it to redraw. And I hit run. And I see the time series for blue, which was chemical species Y, and the time series for red, which was chemical species X. Let's see if people were able to get that to work. Somebody says that it crashed. Are you running this, Stephen? Are you running this on NanoHub, or are you running it locally on your computer? Okay. Are you on a Linux install? Windows. That's concerning. So unfortunately, TJ is at a family emergency, and he is not available to support us this morning. So I apologize. I don't know. Somebody says it's working fine on Linux. Tony, are you on NanoHub or are you on a local machine? Huh. I don't know what to tell you on that one, because is anybody who's running it locally having it work?
There seems to be a problem with the Windows configuration of Tellurium, so I would suggest people who's running on Windows to use it to try NanoHub for now until we can get support from TJ to fix that.
I'm puzzled because I did do this on my local install, which is a Windows install this morning, and it ran. So I apologize. I don't know how to debug that for you today. What I'd ask you to do now is no, Tellurium comes packaged with a CompuCell. So I think that I'm going to have to, I apologize, but I'm going to have to, I'm running 4.2.5 and it works. So if you're running an old version of CompuCell, it's possible there's a bug. So I would ask you, the people who seem to be having trouble seem to be relatively advanced. And so I would ask you either to, 4.2.4 is okay too. So it seems to be confined to 4.2.2. I'm afraid I'm going to have to go on then. Would I ask people to try, if you are having trouble on your local install, please try running it on Nano. And we'll come back and everything that I'm covering now will be covered again by Josh in the afternoon. And then we can have some time to do some one-on-one debugging with you. And I apologize for not keeping everybody with me. I understand that that's frustrating. Josh, if you can reach out and try to help people in a breakout room if necessary, that would be fine. People who did get it to run, did you see the output that I'm showing here?
Professor, I tested in Windows and also in the Mac. In Mac works fine, but in Windows doesn't work. Okay,
then I'm afraid we're going to have to ask people to work on the NanoHub version for the moment.
Does it related to call the Tellurium to install it on the Windows before, or it doesn't need it?
I can't do that debug now. I'm sorry.
Okay.
It should install Tellurium automatically, but I can't. I don't know what the install command would be. It should be probably from Tellurium, import Tellurium as TE is my guess. Giuliano, can you look at that and see if you can see whether there's a quick fix? Again, I'm puzzled because it works fine on my 4.2.5. And so I'm going to keep going here and ask you to follow along in NanoHub if possible for the moment. And then we'll try to debug during the break. Okay, so somebody else says it works fine on 4.2.2. All right. I'm sorry that we don't have TJ here because he's the one who would be able to. Okay, so you're also 4.2.5. So it does seem to be local to 4.2.5. And my guess is, again, if you go to the top of your code, you could try typing here in line 2 it would actually be import tellurium as TE. I can't guarantee that will work, but that would be my 0th order guess for a fix. Again, I can't try it out here on the screen because I don't have the because it's already installed. It won't work. I can't reproduce that. All right. So now let's come back to the tellurium code. The things that I'm going to show you, you can mostly do. Why don't you just not worry about running it for the moment and follow along while I walk through the tellurium code, and then we'll debug it later because we have only half an hour, and I want to make sure that I introduce the basics of how tellurium works. Okay, sorry about that. Okay, so what I want you to look at now is this code between lines 4 and lines 15. As I say, if we'd been lucky, we would all have seen this 2 lines going together. Life is never so easy, I guess. And this is actually running a chemical reaction. The chemical reaction is that X is transforming to Y with first order kinetics at rate B times X. Y is being transformed back to X at a rate C times Y. If we do some simple algebra, we find out that the equilibrium ratio of X over Y is C over B. And so the first thing that we could do is a teeny exercise, and apologies to those of you who it's not working for, would be to change the values of B and C and see what happens. The default here would actually be C equals 0.1. Hit save. I'm going to run it again. If I run it with those values, I see X goes down much more than Y. And so I can change the equilibrium concentration. And let me come back to the code for a second. Okay. So given the time, I think I'll let you play with that on your own. But even in this very simple model, if I change the values of B and C and rerun, I will see that I'll reach a chemical equilibrium. And the value of my 2 species, the concentrations of X and Y at equilibrium, will be different. It's important to note, just as terminology, that X goes to what's called a steady state value. You'll notice that the red comes down and then reaches a constant value. That could be called an equilibrium value in chemistry and mathematics, a steady state value. The blue comes up and reaches an equilibrium value. If X and Y, if B and C are the same, then my equilibrium is an equal amount of the 2 species. If my values are different, I have different equilibrium. So I want to talk a little bit now about antimony mole specification. Antimony's basic idea is chemical reactions. And so you use arrows the way you do in a typical diagram in a biological network. CompuCell, and we're not going to do this right at the moment, you'll see it in the afternoon, references all of the variable names, parameters, and also the rates, E0, E1, and E3 in this, are all available to you as dictionary entries. So if I define an antimony model, which turns R into Q, and I type self.sbml model dot square bracket Q, it will give me the value of Q. If I type self.sbml.model square bracket E0, that will give me the rate of transformation, the value K1 times S. If I type self.sbml.model and a parameter value, K1, it will give me the value of the parameter. Those can be read and written to, just like any other variable. And so I can now connect my Python level to my Antimony level. I can either run the model what's called free floating. That's a single model that runs for the whole system of the model, self.sbml model, which belongs to my entire simulation, or I can load that model into individual cells and run it one version of each model in each cell.
Can we change those values during coding in the simulation, the steppables?
Yes, the whole point is that the simulation that you've written here will define initial values of the parameters and the variables, but I can set self.sbml.model variable equals 26, and that will overwrite the value in the Antimony model. So yes, you can change them. You can't change the rates because those are derived quantities, but the parameters and the variables you can write freely from CompuCell level Python in the steppable. That's exactly the point of the integration.
Oh, great.
So now let's write an exceedingly simple simulation starting from this. If you can write this, you can write anything. Okay. And again, I apologize to the people who can't run it, but even if you can't run it, you can write the version here. So let's take an extremely simple model. This will be a viral infection model. We start out with T cells that are uninfected, and the rate at which they become infected is beta times T times the amount of virus, and they become I1. And so let's actually write that. We're going to first show it to you, and then I'm going to ask you to write it. T goes to I1 at a rate beta TV. That's a rate law. How do we interpret that? We write E, first we put everything inside these model string triple quotes. That just tells us, and we assign that to a string variable, model string. Here, in this example, it says model instead of model string. This is just the name of a string variable. Double slash inside of an Antimony string is a comment. So here, equations is just an annotation saying that I have an equation. Parameters is just an annotation saying that I have a parameter. Initial conditions is an annotation. Semicolon works like a break. It's essentially equivalent to a new line. I can use that freely. I recommend ending every line with a semicolon. So that's the syntax so far. Double slash, comment, semicolon, break. And Antimony writes everything that you do pretty much in terms of concept of chemical reactions. And so I take this diagram, which is T goes to I1 at a rate beta TV. I give that rate a name, call it equation 1, E1, colon. And then I write T goes to I1, and that's a minus sign and a caret, semicolon, the rate law, beta times T times V, semicolon. Notice that I'm destroying T and creating I1. And so that means that this one equation here implements two ordinary differential equations that are balanced. You really don't need a tellurium tellurium. There is code. Twit does have code to paste in the structure for tellurium for loading models, but all you have to write is model equals triple quote and triple quote, and then type the arrows. Okay, so our rate equation here that we want to type, and I want everybody to do this together now, instead of what we've got here at the moment, J1 and so on, we're going to type E1 equals this. Let me actually switch to this. I'll show you the result. Let's see, which one is it? Okay. This is the version that we have. E1 colon T1 goes to I1 semicolon rate. And then we have to specify all the parameters. We have to specify the initial value of T here. And I've done that in 2 steps. I've said T0 equals 1 e to the 7th, T equals T0. I should have written probably just T equals 1 e to the 7th. It's initial value. The initial value of V and the value of beta. Of course, most things will be more complicated. I'll have multiple equations.
I'm wondering how anatomy language is kind of interpreted. So we are defining the equation first, which the beta is not defined yet. Then we assign the parameters to that, so it doesn't give an error, right?
So Antimony is not a programming language. It's a model specification. And so technically what happens is that this string is parsed and is compiled using a JIT compiler to create an executable. This is what's called the declarative language. And I don't want to go into too much detail about it. The order in which you declare things matters only in certain respects. For equal signs, the order matters. T0 first, T equals T0 matters. It's also important that equal signs are only executed once during parsing. There are a variety of issues that will come up. I'd like to skip over the technicalities of Antimony details and refer you to the manual, because most of them are not things that we're going to run into. Antimony allows a lot of very complex modeling. Mostly we're not going to do that because we'll use the Python capabilities of CompuCell to do that. And again, we'll come to that. We will go over those issues. If we wanted to actually have a complicated chemical reaction, 3 molecules of A goes to plus 5 molecules of B go to 1 molecule of A, 2 molecules of C, and 9 molecules of D. Antimony will handle stoichiometry of complex chemical reactions automatically. I can write E2 colon 3A plus 5B goes to A plus 2C plus 9D, semicolon, and here the rate is mass action, K times A cubed B to the 5th, like that. And one thing that I have to watch out for is that in Antimony, the up caret is equivalent to the 2 stars for exponentiation in Python. So the comment character and the exponentiation character are different in Antimony and Python. This single arrow diagram for chemical reaction would generate 4 ordinary differential equations, 1 for the concentration of A, 1 for the concentration of B, 1 for the concentration of C, and 1 for the concentration of D. As I mentioned, when we're going to do regulation, very often we don't care about the things that are producing the species that we're regulating. And so you can have a dangling arrow. If I have a constant source of A is produced at a constant rate A, I write that E1 colon nothing goes to A at a rate K. That means A increases linearly in time. If I have first order decay kinetics, A goes to nothing at a rate K times A, I write E2 colon A goes to nothing, semicolon rate K times A. So if you're used to ODEs, the arrow diagrams may seem a little bit, take some thinking about. If you're used to chemical reactions, writing them is very easy because you write everything in the way you're used to writing them. You can write ODEs explicitly. T accent, T apostrophe is equal to beta times T times V, creates that ODE. I can write exactly the same thing with nothing goes to T. Mathematically, it gives me the same equation. Okay. If I have the same chemical species represented multiple times, antimony will automatically sum them. So if nothing goes to T, T plus V goes to something else, then I will get nothing goes to T at rate K0 in my T equation. T plus V goes to something, destroys T, minus K1 TV. And so I can convert back and forth. Antimony will automatically sum all of the different terms involving each species. Again, it makes it very easy to build modular models, much easier than writing ODEs. And in class, I often will do an exercise where I will show a paper, for example, by John Tyson, where he's written all the chemical net reactions in terms of ODEs. And I'll ask them to say, what was the network that generated it? And you cannot go backwards from the ODEs to determine what the regulatory signaling or metabolic network was. It's not possible. You lose information. You can always go forward from the arrow diagram to the ODEs, but you can't go backwards. And so the reason that I particularly like antimony is that it's a model specification that preserves the biological information. Okay. You have to specify all of the parameters and initial values, and you do that simply with equal signs. Beta equals 6.2 e to the minus 5. V equals 10,000. T naught equals 1 e to the 7th. And here again, there's this double line where I've set T equal to T naught. That is about all you need to know about writing network models. Antimony allows you to do complicated things like events. You can say at time such and such, do this. You can say if something is bigger than something else, do that. But typically we'll do that in Python instead. However, you can do it. Okay, so what I'd like people to do now is take this code that I've got here, the one that I started out with, this one, and make this transformation. I want you to implement this viral infection model. So we started with this code here. All of these models, by the way, are available in the models directory, but just take by hand, you can go in and type, instead of J1 goes to this, we can type E1 colon T goes to I1 semicolon rate beta times T times V. Get rid of J1, J2. Okay. The parameter, there's only one parameter, beta equals, scientific notation works, 6.2 e to the minus 5. You don't need C. Initial condition. Here I've used the 2 lines. T0 equals 1 e to the 7. And then I set my value of T equal to that T0. So why doesn't everybody try doing that?
Is this sensitive to the capital letters and low capital?
So in Python, capitalization matters a lot. Tellurium does not care about capitalization.
Okay.
However, when you use your key for your dictionary entry, it does matter. So when you move into CompuCell, you have to remember if it was capital X here, it has to be capital X here. Okay. And now why don't you figure out what you have to do to display it? It says variables. We're going to have to change my variables line. I'm going to be displaying T and I1 instead of X and Y. So everybody should be able to do that, I hope. And then as usual, you have to hit save. And now I come back to player and I'll stop and I'll hit run. And as usual, I'll have to joggle my screen a little bit. Oops, I had a typo. Global parameter V. Okay, it didn't reload the right. Okay, sorry, my mistake. I have to move it. I have to launch this now. Open the player.
You forgot V.
No, there is no V. Did I not? No, sorry. Did I? Yes, sorry. Thank you.
Included V in the equation, but you have not mentioned the value.
Thank you. I have it here and I didn't type it, right?
Yeah.
Okay, thank you. Well, so appreciate you. Also in Antimony, okay, V equals, but I have 10,000. Okay, Antimony doesn't care about, unlike Python, it doesn't care about indentation. Thank you. Let's see whether it works now. There we go. Are people able to get that to work? Looks like everybody got it to work faster than I did. Yeah, okay. And I strongly recommend using the save as inside of Twit to create new versions of things as you go. I didn't do that here. Okay. So you should have gotten something that looks like that. Okay, let's make a chemical reaction. A plus B goes to C. Why don't you try doing that? I'll do it in parallel. I want you to think about doing it on your own. I've actually got the answer here, but why don't we do that together? I gave it a new rate law name. I called it R1. A plus B goes to C. I shouldn't really talk it through because I want you to try to do it on your own. A times C. I have only one parameter, K. I set it equal to 1. And I have to set my initial values. A, I'll start with no A. Set B equals to 10. And I understand that these aren't meaningful parameters, but they're just to play with for the moment. And I have to set my initial value of C. Spacing doesn't matter. Like that. And now I'll plot A.
Should it be B
B.
and line 6, K multiply A multiply to B? Oh.
Yeah, thank you. And C, I need to do C. I want to plot C, colon, make it red, blue, make it green. Okay. Try that. Notice nothing happens. Why is nothing happening? What was my chemical reaction?
It's A0.
A is 0. There's no substrate, so I can't have the reaction. Nothing happens. So now let's go back, and we won't, since we're going to run out of time. I don't want to spend too much time on this. But now let's make A not 0. Let's make A initially 1. Okay, make A 1. And now given that I have to re-joggle the screen, if you use the stop and then the step function, then you can joggle the screen, get it laid out again. And now I can run it. And now you see A comes down, B comes down, C comes up. When I run out of A, my reaction stops. What if I make A equal to 10? If I make A equal to 10, do it again. Crash on me. That's a good redraw. Now I've decided to die. All right, try again. Why don't you play with that a little bit while I'm getting it relaunched? Which one was I running? I was running simple chemical reaction. This was 0, so I need to open it and twit it. Okay, let's make A 10 and we'll go from there. And now we come back, stop, step, layout, run. And now we have a complete reaction. All right. So you can play with that and you'll get the things that you would expect for a simple chemical reaction. Why don't you try, and this will be the last thing we'll do for this module, why don't you try implementing A goes to B at a rate proportional to A, B goes to C at a rate proportional to C, C goes to D at a rate proportional to C. Start out with no B, C, and D, and start out with some amount of A. So again, the step is write each chemical reaction as an antimony reaction, define initial values for A, B, C, and D, define the rate constants, and then put in the variables line A, B, C, and D. And let's do that as our last exercise for today. I'm sorry about the problems running antimony on your local machines. And we'll see what we can do about that during the break. But I hope people are able to at least get this to work. Really writing reaction networks isn't any harder than this. I guess I can do it myself while we're doing it. R2 colon C colon KB and CB 3. C, B, semicolon. Cut and paste does work inside of this. So if you want to save time, you can cut and paste. Okay, I got my rate laws. A goes to B. The spaces don't matter, but it makes it look nicer if I have the spaces. A, B, A will start with 10, B will start with 0, C will start with 0, D will start with 0. And then I have to plot D as well. Orange. Did that work for people? Somebody asked to be put into a breakout room. You could help them, please. All right. So really, that's about as complicated as it gets. I hope that wasn't too intimidating. And I apologize for the bug in this 4.2.5 install for some people on Windows. But you can see now the rates of the chemical reaction. And one can play now with there are a whole series of exercises, which I give you as homework, where you can explore how these chemical reactions change as I vary the rate constants. We could make this a closed reaction, in which case I'll equilibrate. And I've got a sort of a fun model where I have bistability, autocatalysis. And the nice thing about autocatalysis is it leads to bistable states. You can explore bistability. Bistability is very important for switching behavior, where you want the cell to differentiate, or you want the cell to either decide to do something or not do it, depending on the value of an input parameter. And so those are some simulations that you can try on your own. And I'll let people try that independently. So let me do my module questionnaire. I appreciate people's patience with the issue with CompuCell. I hope that people on NanoHub, that's one reason we like the NanoHub deployment. For all of its annoyances, at least we know that if everybody's using the NanoHub deployment, everybody's having the same experience. And so I'm going to do my module questionnaire. If you want to play with the bistability model, that actually represents a gene regulatory network where you have a molecule that turns on its own production. So it's a transcription factor that is a transcription factor for itself. That model you have to interpret. This arrow, the A promotes A, you have to interpret. You could do it with messenger RNA or not, but you have to think about what it means. There's a standard Hill function form for that. And you could write that Hill function form and implement it in a very simple way. Nothing goes to A promotes itself with a Hill function form. A goes to nothing, decays. And when you write those three equations in the little example we've given you, you'll find that you can do bistable gene expression. And so that's where I'm going to stop for now. So there's a question. Says CC3D suppose these processes are inside a cell. So CC3D allows you to make a choice. In the case that we're running here, we have loaded a single copy of that model to the entire system. So that model is running globally in the system. However, if I have, and I didn't create any cells in the simulation, so there are no cells to load it into. However, if I had 1,000 cells, I could run 1,000 copies of a gene regulatory network, and each gene regulatory network could be different. Each gene regulatory network could be responding to the environment. That gene regulatory network could be changing the behavior of the cell. When the behavior of the cell changes, you can load different networks. So you can actually control with very fine granularity what you're doing. You can have multiple models inside a single cell. You can load and unload models from a cell. You can change the parameters in each cell independently. You can have multiple SBML, multiple antimony models running inside each cell at the same time. You can run the models either deterministically with ODEs, as I've shown it, or stochastically with Gillespie algorithm. And you can do those. You can have some models run deterministically and some models run stochastically at the same time. There's a lot of flexibility. I wanted to show you the very simplest thing, so that I hope it didn't scare too many people away. It looks like I scared 1 or 2 people away. Yes, if you write PBPK models or other kinds of pharmacokinetic models, they're written in exactly the same way. So if I wanted to model transport with compartments, I can do that. SBML, I keep saying SBML because it converts to SBML. Antimony allows you to define compartmental models. And I can have a model of the body with organ systems, and then I could have my CompuCell be a part of the tissue like the liver and run individual metabolic models inside each cell in the liver. So yes, that's pretty common to run a PBPK model in the whole system to represent the movement of a drug or molecular species in the body, then pass that to the individual cells and have them do their metabolism or respond to the drug. So if you wanted to do a model, for example, of drug dosing for chemotherapy, you'd do it that way. Any other questions?
Professor, regarding some functions like load A and simulate that it is possible to work with from the Python. So I don't see that it is used here in the code. So how does CompuCell works with that?
That's exactly what I said. I asked you to not ask that question. I'm teasing you because the whole afternoon, Josh will be teaching you how to do that.
Oh, okay.
We created this template model so that people could just write the antimony and see it display, and not have to worry about loading and compiling things. So in the afternoon, we will show people all of the details about how to write these models, load them, change the settings of the integrators and everything else. But I wanted to give people something where they could immediately say, I have the reaction, molecule A plus molecule B gives me C. How do I get it to work in CompuCell? And so that was the focus today. It's a very good question, and it's exactly what we're going to do later. Okay, any other questions before we break? We're running a little bit late. Why don't we come back at, let's see, we were supposed to have stopped at 12 o'clock. It's now 12:10. Why don't we take a 10-minute break, and can we start back at 12:20? Is that all right for people? And for 12:20, we're going to be doing the hackathon discussion. So if people have previously did hackathon presentations, if you're willing to give us a little update on what you're thinking about your hackathon ideas. If you're somebody looking for a hackathon team to join, please speak up. And I hope that the people who were not able to speak on Tuesday are going to be ready to present their ideas. Okay, so I'm going to, I hope that people can, maybe during the hackathon session, we can have people work trying to make sure that everybody can run this. If we cannot get the 4.2.5 issue solved,